Hello children, hope you all are fine. Now today we will start the structure and development of ovule and female gametophyte. In this we have to study about gynoecium. Gynoecium consists of megasporophylls. Mega means large, sporo means spore, phylls mean leaf-like structure, which collectively known as megasporophyll. It is also known as carpel or pistil. Each pistil consists of three parts. First is stigma. Stigma is dish shape or like a platform like structure which serves to receive pollens after pollination. Next one is style. This style is slender, hollow, elongated tubule which provide the path to the pollen for fertilization. And the last one is ovary. It is basal swollen part that internally contains one or more ovules or megasporangium which produce megaspore and after maturity to form embryo sac. Now structure of ovule or megasporangium. So the ovule is attached to the point inner wall of ovary called placenta. Ovule has major parenchymatous structure that is called nucellus, which is surrounded by a covering called integuments, leaving the opening. This opening present at the apical end, which is known as micropyle. The base of ovule or the opposite part where integuments arise is called chalaza. The point of attachment of the body of ovule to its stalk, which is known as hilum, and this hilum is attached on the base of funicle. Sometimes funicle is continuous beyond the hilum. It is clearly present in anatropous ovule. If only one integument is present, it is called unitegmic. If two integuments are present, it is called bitegmic, and so on. Okay, now we will study about development of ovule. How it is developed. Ovule are developed at the cavity of ovary. It arises as small mound of homogeneous tissue in the inner wall of the ovary, and then it form the central part that is called nucellus. This way the development of ovule takes place. Now, formation of megaspore. In the micropylar region, this region of nucellus, a single cell differentiated from other cells called primary archisporial cell containing big nucleus and dense cytoplasm. This cell periclinally divided to form PPC and PSC. PPC means primary parietal cell and PSC means primary sporogenous cell. Now, primary parietal cell undergoes few division where sporogenous cell gets embedded in as a nucellar mass. Now, sporogenous cell act as MMC, like called megaspore mother cell. It undergoes meiosis and form four haploid megaspores. This is called megasporogenesis. Out of these megaspores, three gets degenerated to provide nourishment for this one remaining megaspore. Only one megaspore will be functional in the ovule, which gives rise to female gametophyte. So, a megaspore is haploid structure which represent the first cell of female gametophyte. Now, formation of female gametophyte. This is the embryo sac develops from megaspore. It is monosporic megaspore in the ovule which gives nourishment and become in the larger size. Megaspore successively divide in three mitotic divisions to form 8 nuclei. All nuclei are haploid and this is strictly nuclear free. Now, this 8 nuclei in this 3 nuclei at the micropylar end undergo cytokinesis to form egg apparatus. This egg apparatus contains synergids and egg cell. Now, other 3 nuclei at the chalazal end undergo cytokinesis and form antipodal cells. Now, remaining 2 nuclei present in the center does not follow cytokinesis and they are known as polar nuclei. Now, a typical embryo sac contains 3 plus 2 plus 3 arrangement. 3 nuclei plus 2 nuclei plus 3 nuclei, it is equal to 8 nucleate. But if we count cell, so 3, 1 and 3, that is 7. Therefore, a typical embryo sac in any angiosperm is known as 7-celled 8 nucleate. Okay. Now, what is the structure of embryo sac? This is the structure of embryo sac in the anatropous ovule. It contains synergid. Synergid is the structure or two cells which present at the micropylar end. In this, egg cells are also present which form an apparatus called egg apparatus. A mass of finger-like structure projections present in the synergid. This is called filiform apparatus, which was discovered by Haberman. It is made up of polysaccharides, which is called cellulosic microfibrils. It guides this filiform apparatus guides the entry of pollen tube by secreting some chemicals for the growth of pollen tube towards embryo sac. Now, egg This egg shows common wall with the two synergids and a central cell. This egg cell lies above the synergid, which is covered with the pectocellulosic wall. Okay, now central cell. Central cell lies in the center of embryo sac. It is the largest cell of embryo sac, remains connected to the egg, synergids and antipodals by plasmodesmatal connection. Now, these antipodals helps in the attachment with the embryo sac, and this whole structure will help in the fusion to form secondary nucleus, which participates in triple fusion. That's all for today's topic. In the next topic, we will study about pollination and the further process present in this chapter. Thank you.